Why is entropy of mixing key for cell membrane transport? Imagine molecules as tiny travelers moving across a busy city. They don't just move randomly. Their movement is driven by a fundamental principle called entropy. Entropy measures how disorganized or random a system is. When molecules mix, they increase the system's entropy because there are more ways for them to be arranged. This increase in disorder is what makes mixing spontaneous and natural. In cells, molecules like ions and solutes are often on one side of the membrane. They want to spread out and become evenly distributed. This spreading out increases the entropy of the system, making the process thermodynamically favorable. So, molecules tend to move from areas of high concentration to low concentration without needing extra energy. This is called passive transport, and it relies on the entropy of mixing to happen naturally. Think of it like pouring cream into coffee. The cream naturally spreads out and mixes because it increases the overall disorder of the drink. Similarly, molecules move to maximize their entropy, which is why they tend to spread out across the cell membrane. When molecules reach an even distribution, the system is at maximum entropy, and no more spontaneous movement occurs. The entropy of mixing also influences how molecules pass through specialized channels in the membrane. For example, ion channels are shaped like funnels. As ions move through these channels, the changing space along the channel creates an entropy gradient. This gradient acts like a push, helping ions move along without needing energy. It's like a slide that guides molecules smoothly from one side to the other. Even in active transport, where energy from molecules like adenosine triphosphate is used to move substances against their concentration gradient, entropy still plays a role. The overall process must result in an increase in total entropy, including the surroundings. This means that while the cell uses energy to move molecules, the total disorder in the universe still increases. Membranes themselves are affected by entropy too. The arrangement of phospholipid molecules in the membrane can change shape and flexibility based on interactions with proteins or particles. These changes in membrane structure are driven by conformational entropy, which influences how membranes bend and how substances are transported. In essence, the entropy of mixing is a key factor because it explains why molecules move across membranes on their own. It provides the thermodynamic reason for spontaneous movement, helps generate forces within transport channels, and even affects the physical behavior of the membrane. Without this principle, many of the processes that keep cells functioning smoothly would not happen naturally.